Hi guys, we're Group CTH. I'm Chichi.
And I'm Logan.
And we're here to discuss the results of our Brachypodium gene investigation.
So what did this investigation contain? Well, we began with an unknown Brachypodium distachyon gene, and we were tasked with determining its function. To do this, we searched for homologous DNA sequences. Then we searched for the protein family classification. And through literature review, we were able to determine the function of our protein family. Next, we looked at gene expression results from various tissue locations within Brachypodium. Next, using this preliminary data, we designed an experiment involving caffeine to inhibit growth, and we analyzed differences in gene expression through RNA extraction, RT-PCR, quantitative PCR, and then compared our results to learn more about our gene.
We used our known Brachypodium gene and ran it through a software, which gave us a prediction of potential genes. So from there, we found that our gene has a single exon with no introns at all, and that our gene had about 590 amino acid protein sequences. And then using these sequences, we were able to run a blast. And from the blast, we got a lot of sequences that were closely related to our gene. However, a lot of these sequences were either uncharacterized or hypothetical or were just unknown. So we had to perform more research to figure out our gene and its function. Using a blast, we trimmed it down and we were able to construct a phylogeny tree. And from the phylogeny tree, as you can see, our protein, our gene is highlighted in red. And our closest relative is the Oryza sativa japonica, also known as rice, as you can see in the phylogeny tree. It's the one above it.
All right, so our next step was taking the closest relative to our protein of interest, which was the Oryza sativa, and using Interpro to learn more about it. We determined that it was a member of the O-fucosyl transferase family, which is located on the Golgi apparatus as well as the ER, so likely involved in macromolecule modification. It has the function of binding calcium ions and also transferring a group. And it seems to be implicated in carbohydrate metabolism as well as fucose metabolism. Literature review revealed that this enzyme transfers an O-fucose group, which is similar to glucose or a type of monosaccharide, to growth factor-like domains. It's also been shown to be important for cell wall adhesion through pectin modification, which has led us to believe that it could be implicated in plant growth, especially through cell wall adhesion and regulation. This figure from Soto et al.'s 2019 paper shows this red triangle fucose being added on top of cell wall polysaccharide, again reinforcing the importance of this enzyme in cell wall adhesion. Next, we took a look at the gene expression analysis within the Brachypodium EFP browser. We found that the plant tissues with the highest levels of gene expression were young and growing plant tissues, such as the young spikelet, the young grains, the middle-length grain, as well as the germinating grain and the young seedling. So plant tissues that are actively growing, which again reinforced our hypothesis that this gene is involved in plant growth. Next, we took a look at the Hazen lab data from mRNA abundance from various tissue sites. We performed statistical analysis between the mRNA abundance at these different sites, and we found statistically significant differences between each of the groups, with the highest mRNA abundance being found in the stems. Over here on the right in the box plot, you're seeing the same information. This shows a low standard deviation between the mRNA abundance in each tissue site, with the highest mRNA abundance being in the leaves. So using our preliminary data, this led to us formulating a hypothesis that stated if our gene is involved in plant growth through cell wall adhesion and carbohydrate metabolism, then inhibiting plant growth will decrease gene expression, especially in the stems.
Since our gene was abundantly expressed in the stems, we came up with the experimental design to inhibit the growth. So we treated our plants with about 15 microliters of an 8 millimolar concentration of caffeine to test the different gene expression levels. So we had 6 plants, 3 of them will be the control, and then 3 of them will be the plants that we treated with the solution that included the caffeine. So from there, after treating the plants, we let it sit for about 3 hours, and then we froze it down using liquid nitrogen. And then from there, we did an RNA extraction, and then ran a PCR, and then we did a PCR analysis, which gave us our results, and our results basically supported our gene hypothesis.
Right, so our gene hypothesis was that the caffeine would inhibit gene expression in our treatment samples, and you're seeing just that here. The control samples had much higher levels of gene expression than in the caffeine treatment groups. Unfortunately, we only obtained cDNA from one of the treatment groups, so we couldn't do proper statistical analysis to ensure that the differences were statistically significant. We also see lower levels of gene expression in the G treatment compared to the wild type control. In the box plot to the right, you're seeing the same information. You're seeing a wide range in the relative expression within the control samples. You're seeing just a single sample for the treatment and also a single sample for the G treatment as well. So our results are somewhat inconclusive because we obtained cDNA for only a single treatment sample, but these results do support our gene hypothesis that the caffeine decreased gene expression.
Throughout this entire lab and our experiment, we did learn a lot about our gene. We learned that it contains a single exon and has about 590 amino acid protein. We learned that it is indeed a member of the O-fucose transferase family, and which is very important in plant growth through carbohydrate metabolism. And then we learned that it showed the highest gene expression in plant stems. So from that, we ran our experiment to compare controls and treated plants. And of course, the treated plants showed decreased gene expression in the stems compared to the control plants. So which kind of supported our hypothesis, although results may not be the best.
So in conclusion, we've gone through the initial stages of gene annotation. We could contribute our data to the scientific community by completing the annotation on the basic local alignment search tool or other publicly available sites, so other researchers could learn more about their gene of interest if it's closely related to ours. Future experimentation would involve repeating the RT-PCR and the quantitative PCR to ensure accurate results. We would also need to include more plant samples to ensure adequate sample sizes, and we could also test the changes in expression of our gene of interest under new conditions as well. Here are our references. Thank you so much for a great semester to Dr. Verma and Ahmed, and have a good day.
Thank you. We hope you've learned a lot from our presentation.